Okay, we are all set, and I'll begin in 3 seconds. So, in the third episode of the Combine Guy podcast, with us we have Mr. Roshan Rao, a PhD scholar at the University of California, Berkeley. Roshan has experience in deep learning. He currently employs deep learning to model proteins from their amino acid sequences. Thank you for joining, Roshan.
Yeah, thank you very much for having me.
Yeah. So, Roshan, we basically discuss your PhD experience here. So, to begin with, you started with a bachelor's in applied mathematics and computer science, and then you had interned at the Space Telescope Science Institute. Now you're working towards your PhD in protein sequence models. So, how did you begin with this journey?
So I had actually initially planned on going to medical school,
Hmm.
but I had picked up an interest in computer science about the summer before I started my undergrad. And so I ended up taking some an intro to computer science class in my first year as an undergrad, in addition to organic chemistry and some of the prerequisites for medical school. And so about towards the end of my first year, I realized that I was a lot more interested in computer science than I was in going further into medical school. And so I sort of switched tracks from computer science or from medical medicine into computer science, and but I still had that chemistry and biology background. So some of the research that I started doing in undergrad was also related to computational biology.
Hmm. That is interesting. So, was this transition a bit easy because you say that you have some background knowledge in chemistry and biology? So, was it helpful and easy to transition from CS to CompBio?
I think it was helpful to have taken those classes in organic chemistry and basic biology. I think that I don't think it was necessarily easy. Definitely, still there's a plenty I don't know about the biology side of computational biology, and there are people who are much more familiar with it than me, who came at it from more of a real like comp bio background originally, and who took a lot more classes and who spent a lot more time studying those topics. So I think that um, yeah, those classes are always helpful. More classes in biology is all helpful, but important to both take those classes, but also to keep studying and to keep learning more about biology and about what you don't know.
Yeah. So from what you say, it's like you'll have to learn outside academia to survive in academia. That's understood. Yeah.
Yeah, I think definitely as you get into, if you try to transition into a new field, especially as a grad student, you're not necessarily going to be able to have the time to take all of the basic classes that you need to study that field, and so you need to kind of do that on your own in some way.
Mm.
And it helps to surround yourself also with people who know the field better than you. It's very easy to fall into traps of thinking you've discovered something or not realizing that you have some flaw in your setup if you're not familiar with your data and you're not familiar with the biology. And so being surrounded by people who know that information a lot better than you is very helpful.
Mm. So yeah, we do need guidance from other PI and staffs. Agreed. So what were the challenges that you had faced during this journey, and how are you trying to overcome this?
I mean, I think one of the primary challenges originally was really having a very limited sense of Um, what was reasonable? There are a lot of questions when it comes to proteins about um, how similar your training and test sets could be. Um, what uh, what constitutes a meaningful improvement? What constitutes what are the kind of biases in the data? What are the tricks that you need to think about? What are the pitfalls in your analyses that people care about? And so when I was starting out doing this, all of the people I was working with were very much on the CS side, and had a lot of good insight and experience on how to design good models and how to think about modeling data from a computer science aspect, but didn't have that the knowledge to know, what does this data really look like? Where are the biases from the biology aspect of things? And so part of fixing that has been me taking classes, me reading papers, and trying to understand that. Part of it has been trying to work with people who do know that more, who are more familiar with um, the biology component. And so I think that's been one of my biggest challenges, and the biggest things that I've been trying to address is to uh, get more of that mixture perspective. So I can bring in the CS uh, like deep learning background, and then someone else can bring in the other background necessary.
Hmm. So, so for someone with knowledge in biology, but he or she is new to computing, what do you think they should start with? Like, what would your suggestions be?
Yeah, I mean, I think it depends on what you're interested in. Like the field of computational biology is very large. You have, everything from modeling proteins, even within modeling proteins, there's a whole set of topics you could focus on. But there's also things like uh, like genome, genetic modeling. There's aspects of like predicting of medicine, more on the clinical side. There's a
Hmm.
whole set of computational biology that's has less to do with molecules and more to do with like patient predictions and outcomes. And all of these are very different fields. And someone who works on, for example, medical text processing is going to have a very different set of problems and a very different set um, of knowledge that they need than someone who works on, say, variant calling with from DNA sequences. And so I think that you know it depends on where you are in the journey. I think if you are like a PhD student, the answer is different from if you're an undergrad. If you're an undergrad, I just recommend, taking the basic classes and focusing on learning the simplest like intro to comp bio 101 like type of classes, because you have so much time and uh, so much of the field to explore, and you don't really know even the things that you don't know yet. So it's kind of like you need to take these wide breadth classes to really understand what the field looks like. I think as a PhD student, you might need to be more focused. Maybe you can take something that's like explicitly related to what you're studying, read the papers that are exactly in the field that you know, and that can help you sometimes pick things up more quickly and in a way that's more related to your actual research. So I think that is an answer that's really dependent on where you are in the process.
Hmm. So, identifying your pluses and your backlogs would help you. Understood. So, uh, we were speaking about PhD, and right now there is a rise in the number of MOOCs for ML and DL. and I think it is being democratized very heavily. So, in such a situation, do you think working in a company that employs these technology would be better than pursuing a PhD in the same?
I think that they are very different experiences. Um, when you are pursuing a PhD, the goal is not to get a good understanding of how deep learning models work, really. It's to produce genuinely new research. And so sometimes companies are focused on that, but when you're employing these in companies, you are not necessarily, you're almost certainly going to get good at engineering. You're going to get a good understanding of how to write these models and maybe how to debug these models. I think you maybe don't get as much of a sense of broader questions like um, what model is actually fit for my data. If this model isn't training successfully, what can I understand why that's happening? Some of these things you do get at companies. It really depends on the type of company and the type of work you're doing at that company. There are some jobs that are extremely research oriented, um, but there are also jobs where you are only going to use this in a very superficial manner, where you're, going to get very good at engineering. But I don't know that you're really going to probe some of the deeper questions and really be generating new knowledge about how to model uh, the problem that you're interested in.
Mm. So these can be treated like they are poles apart. Mm. So
Yes,
tell us,
go ahead.
yeah, yeah, go ahead. Sorry.
Yeah, I was just, yeah, it's definitely, it's very company dependent. And you can get a really great job where you are doing something that's very close to PhD research in a company. Those exist, but there are also plenty of jobs where that's not what you're doing, where you're I mean, the focus is also can be very different. Focus on the specific data that the company uses, and a lot of limitations surrounding what questions you can explore based on that data. And then maybe a lot of focus is on data cleaning, or you have like very, very specific problems that you're trying to solve rather than maybe trying to ask more broad and general questions. So I think it's very, very company dependent.
Mm. Okay. So your PhD, is it like totally related to proteins, or you create libraries and work on improving the computational efficiencies of other existing models?
Yeah, so I have done a couple of other projects in my PhD. I initially, in the first year or two, was kind of not sure what direction I wanted to work on, and so did some additional projects. At this point, I'm pretty settled on focusing on modeling proteins. Um, I mean, I think I have created some open source libraries, but I think I put that under, modeling proteins and helping people in the community be better at modeling proteins and understanding how to model proteins. So that's kind of my current focus.
Mhm. So your paper on the protein embeddings. So how did you get that idea, the intuition behind such an embedding?
So, I mean, I think it very much borrows from language modeling, and the idea that um, you have these representations of English and other languages now that seem to be really good for doing things like question answering and completing sentences, etc. And so, if you look at BERT, GPT-3, GPT-2, GPT-3, these models all came out around the time when we were thinking about tape, and which is the protein modeling paper, and we thought it would be a good idea to try these models on proteins, and proteins look a lot like language. They are these 20 amino acids. Each amino acid can be represented by a character, and so a protein kind of looks like a string in the same way that a piece of text does. So it sort of makes some logical sense. Now, actually, there have been some recent papers that draw a lot of connections between BERT models and previous older methods on proteins like POTS models and pseudo likelihood. And so there's actually some connections you can draw between these newer deep learning methods and the older kind of more classical machine learning methods on proteins. And so I think there is both this intuition that that these models worked well in language, and so maybe they would work well in proteins. That has more to do with the fact that just the inputs and outputs look very similar, and so you can pretty easily take a language model and put it on a protein. And I think some of our understanding maybe now is more that actually these models do make a lot of sense on proteins, and they have these strong connections to models that have worked well in the past as well. And so we believe that they should be able to perform well when we look at proteins as well now.
Okay, in that case, is it true that these models have a biological intuition in modeling? Like, do they represent any sort of biological characteristics or uh, something associated with biology and not just model language modeling?
Yeah, so kind of the fundamental thing that you'd think about is that um, when you have, and this is how, so for a long time, people have used POTS models, pairwise Markov random fields, to model proteins. And the idea behind this is that you can uh, you can kind of cheat to learn extra information about a protein by looking at similar proteins in other organisms. So if you
Hmm.
look at the kind of evolutionary neighborhood of a protein, you look at all of these similar proteins in other organisms, you'll find that evolution has changed these proteins to some extent. So you have some amino acids have been substituted for other amino acids, and then some have been deleted, some have been inserted. If you align all of these similar proteins together, then you can get a sense of which amino acids are conserved. So which uh, positions are likely to stay the same throughout evolution. That might tell you if a position is highly conserved. That might tell you it's very important. You generally think that if a position is unimportant, it could mutate to almost any amino acid, and or at least maybe amino acids with similar properties, and it wouldn't really matter very much. If an amino acid is very important though to the function of that protein, let's say it forms part of a binding pocket or something, then mutating that amino acid is likely to break the function of that protein. And so if you look at evolution and you find that some amino acids are less likely to mutate than others, then this can tell you that those amino acids are more important. Similarly, you can look at coupling between positions. So you can say that normally you would expect two positions to be roughly independent of one another. So one might mutate, another might mutate, but if they're not interacting with one another, then you expect them to be basically independent of one another. But if they do interact with one another, then maybe you would expect that only certain pairs of amino acids can be present at that location without breaking that interaction. So for example, if you replace one amino acid with another, and now the side chains between these two amino acids are clashing, then that's going to probably result in a non-functional protein.
Okay.
So you can look at the course of evolution, you can look at what are these statistical patterns that emerge if we look at all of these organisms. And we can look at things like mutual information between positions. And that tells us something about which positions are related to one another. So high mutual information, high pairs of positions or pairs of positions with high mutual information are likely to be interacting in the underlying sequence. So then you can ask another question of like, and so then you can model this by saying like which positions are predictive of other positions. That's what a pairwise Markov random field model does, is it says, you know, which, if I know the value of position A, what does that tell me about the value of position B? If you do this and add some like L2 regularization, basically a prior on that model, that gives you an intuition that, or that gives you the set of likely interacting residues, which tells you something about the 3D structure of the protein. And it and then you can generalize this to like large scale transformer models by saying like, okay, that's basically a language modeling task. That kind of information, like which pairs of amino acids are predictive of each other. Well, that's saying if I mask out a position, which amino acids are informative about that position. It's essentially a language modeling task. And so that's kind of how we draw this connection between pseudo likelihood and these old standard models and the more modern models that people are using these days.
That is interesting. This is the first time I'm coming to know of the biological inference behind language models. So that was interesting and a useful insight. So, how was your internship at the Space Telescope Institute helpful? Like the contacts that you made, and like, did it play any part in you taking up a PhD in CS?
Yeah, absolutely. I mean, that was my first real job in kind of programming and computer science, and I did it in my first year, the first summer of undergrad. And so I had about a year of programming experience when I went into that, and it was a pretty research focused. I mean, the Space Telescope Science Institute is a research organization. It was a research focused internship where we were really trying to build a a method for constructing 3D printable galaxies. And the goal was to use these for blind students who aren't necessarily able to see or who aren't able to see what a galaxy looks like in images. We were trying to find ways to convey things like flux and the composition of galaxies in a non-visual manner. And yeah, it was definitely very research oriented. It's a very research oriented question, kind of HCI, human computer interaction, and modeling. Um, in this context, yeah, and I think that it definitely made me interested in computer science research, and it was directly useful towards getting a PhD because I got a letter of recommendation from my mentor there, and so those types of research internships are very useful towards going towards a PhD, both because of the like mundane kind of letter of recommendations, but also to tell you if that's a line of work that you're interested in pursuing, if that type of work is something that's interesting to you.
So that was an interesting and a new thing. I never knew that people make 3D models for blind people, visually challenged people. So that is something good. And regarding open source technology, you have contributed to open source technology. And right now almost all the models in the deep learning era are being open source. They are publicly let out for use. So do you think this will have an impact, or this will help us move forward as a society together?
I mean, I think open source is certainly very good. You know, there is, I think, very limited benefit to not open sourcing deep learning models right now because they are so easy to uh, recreate. With the exception perhaps of truly very large models like GPT-3 or
Three.
models that are highly dependent on data, where I think maybe there is some competitive advantage still to keeping them quiet. I really just don't think that there is very limited competitive advantage right now to having a private deep learning model, or to having like deep learning methods be private. Um, of course, I'm sure that their I'm sure Google has their own kind of private versions of a lot of the models that they use inside of search, et cetera. And certainly in biology, I don't think these models are good enough yet to really be monetizable and to really have to really, provide a lot of advantage to someone for keeping it private. In biology, I think a lot of the advantage comes from collecting the data, and having kind of those kind of wet lab setups, and that the computational methods are, really useful, and the open source helps drive communities forward. But um, if you do keep it closed source, you should expect to have someone crack it within a couple months, or reproduce your results within a couple months. So I think that both it is beneficial for the community that people are focused on open source, but that also there is not that much of an incentive right now to focus on closed source.
Hmm. So that is one interesting perspective to look at. Thank you for joining us, Roshan. It was good meeting you, and we did gain a lot of insights from your research. Thank you.
Yeah, thank you very much for having me. Great to be on.
We also hope that you contribute a lot to the society and in general to open source models.
Yes, I hope so as well.
Yeah. So I'll stop the recording.